Good morning, everyone. Welcome to UCLA, and thank you for this fabulous opportunity. My name's Khakh. I've been running a lab for about 18 years, 13 of those here at UCLA. In my lab, we work on a type of brain cell called the astrocyte. Astrocytes represent about 40 to 50% of the total number of cells in the brain, which, if you think about it, is a remarkably high number considering how little we actually know about them. So when we started to think about this problem a few years ago, we set ourselves three goals. The first of these was to develop tools to study these cells properly in the adult nervous system. The second was to determine what these cells actually did for the brain in normal function. And the third was to begin a sort of a voyage to try and exploit these cells for diseases of the nervous system. And so what I want to do in the next couple of minutes is to tell you about these three things. So in this first movie, what you can see is a sagittal section of the brain sort of in this axis. And what this shows is that all these astrocytes are labeled green. And the only point in showing you this movie is to make really 2 points. First is that these cells exist at high density throughout the brain. And the second is that we now have the genetic tools to specifically target them and achieve gain and loss of function studies, which means we can do hypothesis-driven work on these cells. In the area of brain biology, a few years ago, we discovered these sparkling signals, which I call the sparkling panorama of calcium signals. These flashes that you can see are, in fact, the elementary communication signals that these cells use to talk to themselves and to surrounding neurons. And we've had a lot of fun trying to understand what these signals do. In the area of brain disorders, I'd like to mention three bits of information. The first is we've been working on Huntington's disease for a few years, and a couple of years ago we found a protein called KR4.1 was downregulated in Huntington's disease. And what this suggested was that this protein, if you could correct it, would change the symptoms in Huntington's disease disorders in mice, and that represents these cells as a target. More recently, in a paper that just went on last week, we found that astrocytes contribute to ADHD-like phenotypes in mice. And in that project, we discovered two targets, a GABA-B receptor and also another target called the thrombospondin 1 receptor. And in a paper published at the end of last year, we found that astrocytes contribute to OCD-like phenotypes in mice. And you can see this mouse is excessively grooming. And this excessive grooming in a mouse models in humans, which is OCD phenotypes that you may see in the form of excessive hand washing. And in this study, what we found was that there was a protein called GAT3 within the astrocytes, which is a therapeutic target. And I think these three examples illustrate that we're making progress in this regard. So to summarize then, our sort of 10-year goal, and I think we're 3 years into this, is to take fundamental biology of the astrocyte and use it to deliver therapeutics and repair for brain diseases. And we're about halfway through the exploration and discovery phase. We want to transition into the evaluation and validation phase, then ultimately use this knowledge to develop and treat major brain disorders. We've got extensive collaborations already, but we're looking for further collaborations with medicinal chemists and technologists. And of course, we're looking for partnerships to help us along this mission. And I think if we were to complete this voyage, we'd be able to develop new therapies for a variety of diseases, including OCD, ADHD, depression, Huntington's disease, and Alzheimer's disease. And so I'd be happy to discuss this further with any of you over the posters. And with that, I'll finish. Thank you for your attention, and see you at the posters later today. Thank you.